Good afternoon. So as this symposium has been organized by Philippe, I thought it would be appropriate to tell you a story which started more than 20 years ago when I was in his lab with the cloning of NF-kappaB. And another reason for that choice is that as we were progressively identifying additional components of that signaling cascade, Jean-Laurent Casanova, who was in Philippe's lab about at the same period, was independently identifying patients carrying mutation in precisely some of these additional components. So this led to a very fruitful collaboration, which is still ongoing, as you will hear. So what I'm going to do, I will tell you a brief description of a number of pathologies which are the consequence or either a complete loss of function of NF-kappaB, partial loss of function. Also something which is less known is the consequence of a gain of function of NF-kappaB. And at the end of our talk, I will report the preliminary results of the analysis of patients that have a partial loss in linear ubiquitination, which it turns out seems to also impinge, at least in part, on the NF-kappaB pathway. So as you all know, the NF-kappaB pathway is almost like a housekeeping function because it's used in about everything. So in particular, it's in the inflammatory response, the immune response, in various steps of the oncogenesis process, and in response to a number of additional stresses. So this is a cartoonesque version of the NF-kappaB pathway to activate the NF-kappaB pathway in response to TNF. So it starts activation of the receptor by the ligand, leads to recruitment of a series of adapter molecules, one kinase and a bunch of ubiquitin ligases, which ubiquitinate the kinase through lysine 63-link polyubiquitin chain, and those chains allow the recruitment of two kinase complexes. The first one is the so-called IKK complex, which binds polyubiquitin through the ubiquitin binding subunit NEMO and two kinase subunit IKK alpha and beta, and the upstream kinase, TAC1, which also recruited to the polyubiquitin chain using the ubiquitin binding ability of TAP2 and TAP3. So then TAC1 activates IKK, IKK phosphorylates the I-kappaB inhibitory subunit, leading to its ubiquitination using this time lysine 48 link polyubiquitin chain, which lead to degradation by the proteasome and nuclear translocation of the NF-kappaB dimers. In addition, the NEMO molecule itself is ubiquitinated. Also, the function of that event is not really clearly understood, possibly by lysine 63 link chain or possibly by linear chain. I will get back to linear chain towards the end of my talk. So one thing I'd like you to remember is the IKK complex is the core element of the NF-kappaB cascade. That's where all the signals converge. And interestingly, NEMO is the only non-redundant subunit in the entire cascade. So before presenting the pathology, I'd just to spend 1 minute about the structure of the NEMO molecule. Essentially, this is a very long dimeric coil with a C-terminal zinc finger. The N-terminal part is involved in the interaction with the two kinase subunits, and the C-terminal is a complex multipartite ubiquitin binding domain that I'll describe also later in my talk, which is absolutely necessary for the response of NF-kappaB to most signals. With a few exceptions, viral proteins such as TAX or VFLIP do not seem to use that C-terminal ubiquitin binding domain. So two things. NEMO is an ubiquitin binding protein, and it also becomes ubiquitinated following NF-kappaB activation. I'll get back to those two concomitant events later in the talk. So the story started when right after we cloned NEMO, and we've been contacted by a consortium of geneticists, including Arnold Munich at Necker, who were after the gene responsible for a pathology called incontinentia pigmenti. So what is it? It's a genodermatosis, it's X-linked, and it's characterized by prenatal male lethality. Female patients are characterized by the complicated skin phenotype, which develops into 4 stereotype steps that I will show you briefly here. That's the first step, vesicular stage. Second step. The third step is hyperpigmentation, hence the name of the pathology, incontinentia pigmenti. And those marks progressively disappear before adulthood, leaving or not area of hypopigmentation with absence of hair follicles and sweat glands. So the analysis of cells derived from IP patients demonstrated that they're completely refractory to all NF-kappaB activating signals. Here I show TNF. We get the same response for LPS, IL-1, and all known signals. The consequence of this defect in NF-kappaB is a hypersensitivity of those cells to apoptosis, demonstrated here by the usage of TNF or TNF plus cycloheximide, cells derived from 2 patients. You see that they die very quickly. Genetic analysis of these patients demonstrated that all cases of incontinentia pigmenti are due to mutation in the NEMO molecule. And 80% of these are the consequence of the same rearrangement between 2 tandem repeats that removes most of the gene. So what could we learn from the IP patients in terms of the NF-kappaB physiology? Of course, it was interesting because it was the first pathology that was due to complete defect of NF-kappaB or a defect in NF-kappaB activity. However, it was not that informative because first, prenatal male lethality does not tell you much. And we already knew from the mouse work that the lack of NF-kappaB translates into death before birth. And the symptoms observed in female patients is not very clear whether they were due to bona fide NF-kappaB deficiency or are the consequence of random X inactivation and interaction between normal cells and the cells that are NEMO deficient. So what would have been useful at that time would be the possibility to identify hypomorphic NEMO mutation that would be studied in male patients that probably would be more informative. So that's where Jean-Laurent enters. And about exactly at the same time, he was identifying patients that showed an unusual association between an immunodeficiency and a syndrome called anhidrotic ectodermal dysplasia that I will describe in a moment. And it demonstrated that these patients, most of them, well, all of them at that time, had a point mutation in NEMO. So this is exactly what we were trying to identify. So the immunodeficiency exhibited by those patients was classical type that can be observed in NF-kappaB as well, already been observed in several mice that have been generated before. So infection with certain number of bacteria, but no viruses. Most NF-kappaB dependent pathways were partially or completely defective. The immunological phenotype was heterogeneous. And as expected, analysis of the NEMO molecule found in those patients, shown here, shows an intermediate phenotype between incontinentia pigmenti, where there basically no NF-kappaB activity, and wild-type cells with that high NF-kappaB activity. So this is exactly the intermediate phenotype, the hypomorphic mutation that we were looking for. So immunodeficiencies is something which is easy to understand in connection with NF-kappaB, but what about anhidrotic ectodermal dysplasia? Well, actually, that pathology was known for quite a long time. It was first described by Charles Darwin more than a century ago, and it was a study of family demonstrating only in men. They had very little hair. They had almost no sweat glands, which means they could not stay in the sun because they were unable to sweat. And they also showed missing or conical teeth. Interestingly, the genes responsible for EDA has been identified before, but this is EDA non-associated with an immunodeficiency. And the EDA was known to be caused by mutations in 4 genes. 2 genes coding for a member of the TNF family. 1 gene, sorry. 2 genes coding for a member of the TNF receptor family, and 1 gene coding for an adapter that interacts with one of the receptors. So what does this have to do with NF-kappaB? Well, as some of you might have guessed already, these three proteins align in a single signaling pathway with the receptor, the ligand, the adapter, and this signaling pathway signals through NF-kappaB, which means that mutation in here, in here, in here, and in here all lead to anhidrotic ectodermal dysplasia. In addition, since NEMO is the most downstream component of the cascade, mutation in NEMO also lead to the immune defect observed in these patients. Later on, a large number of mutations were identified within the NEMO molecule, and depending on their location, they would give rise to three different types of pathology of different severity. The mildest form would give rise to immunodeficiency, indicated in blue. Then in green are the immunodeficiency associated with anhidrotic ectodermal dysplasia, and the most severe forms would give rise to incontinentia pigmenti, which means they would almost completely wipe out NF-kappaB activity. You will note that a number of those mutations are located either in the C-terminal zinc finger, and I get back to that, or in these central elements that I will describe in a moment, which is called NOA. So far I only discussed loss of function of NF-kappaB. Then the next question is, how bad is it to have too much NF-kappaB? As you will see, it's not great either. And this is a work that's been done in collaboration with a group of David Valach at the Weizmann in Israel. And this basically identified a pathology which is due to a hyperactivity of NF-kappaB. It started with the two hybrid screen using NEMO as a bait, leading to the identification of a large molecule of more than 1,000 amino acids called SILD for cylindromatosis. This molecule was known before. Its function was unknown, but the molecule was known to be, mutation of that molecule were known to be responsible for a pathology called familial cylindromatosis, or turban tumor syndrome. This pathology, which gives rise to a large number of tumors on the head and the scalps. Show you one example here. And what we demonstrated is the fact that SILD is actually a D-ubiquitinase, which is specific for lysine 63 link chain. This is shown here, and you see that the SILD molecule, which normally removes polyubiquitin, if you mutate the lysine of ubiquitin to an arginine, there is no activity of the SILD molecule. However, if you mutate the lysine 48 to an arginine, then the molecule is still active. But then if you take a molecule which is derived from a cylindromatosis patient, again, he has lost the activity, which means that the ubiquitinase activity of SILD is important for the pathology that we described. So the interaction with NEMO suggests that it might be connected to NF-kappaB, and in fact it is. If you transfect SILD into cells and measure NF-kappaB activity in response to TNF or IL-1, you see in green that there is a strong downregulation of NF-kappaB activity, which no longer takes place when you use a molecule such as the one found in patients that have mutation in SILD molecule. So this actually means that SILD deficiency results in hyperactivation of NF-kappaB. How does that work? Well, the most likely explanation, which has been partially demonstrated, is the fact that SILD, which turned out to be a NF-kappaB target gene, once synthesized, removes those lysine 63 or linear chain which are attached to the TNF receptor and stabilize the signalosome. Actually, another molecule called A20 apparently able to do the same thing. SILD is the only ubiquitinase which is able to remove both lysine 63 and linear polyubiquitin chains. Just a bit of additional information. René Bernard, who also identified SILD at the same time using a different strategy, pushed the idea a bit further and tried to treat patients affected with cylindromatosis using inhibitor of NF-kappaB. So what he did is to treat patients with mild forms of cylindromatosis with a cream containing acetyl salicylic acid, which is a non-specific inhibitor of IKK. And what he was able to obtain after several months of treatment is a partial reduction of the benign tumors found on that particular patient. So before I switch to the last pathology related to linear ubiquitination, I need to first introduce linear ubiquitination to you, and then present the results of a work that's been done in the lab in collaboration with Fabrice Agou in Michel Véron's lab regarding the ability of NEMO to bind different types of polyubiquitin chains. So what is linear ubiquitin? You probably all heard of polyubiquitin as branch chains, where each ubiquitin is attached to an internal lysine residue of the previous ubiquitin moiety. But about 5 years ago, Kazuo Iwai isolated a complex that he called LUBAC, which was able to elongate head-to-tail linear polyubiquitin chains. And the function of this complex is not really well understood. However, so far, the only signaling pathway where this complex is involved seems to be the NF-kappaB pathway. So this is again the version of the TNF pathway where the LUBAC complex is included. And so far, the only substrates of LUBAC are either NEMO or possibly RIP1, so this is not as clear. So the LUBAC complex is made of 3 molecules, a catalytic subunit called HOIP, two regulatory subunits called HOIL1 and Sharpin. And the function of this complex through linear ubiquitination of a certain number of components of the cascade is to help in the formation or the stabilization of the signalosome that forms as a TNF receptor and probably at other receptors which are NF-kappaB dependent. So I told you that NEMO was ubiquitinated with either linear or lysine 63 chains, but we also demonstrated that NEMO is able to bind both linear and lysine 63 chain. NEMO is part of a small family of 3 proteins, optinurin and abin2, which contains the same C-terminal complex ubiquitin binding domain. And while the group of Ivan Dikic demonstrated that that element of the ubiquitin binding domain called NOA, it's about 30 amino acids, is able to bind with high affinity linear polyubiquitin chains, we demonstrated that the combination of NOA plus the linker plus the C-terminal zinc finger constitutes a high affinity lysine 63 specific ubiquitin binding module. And therefore, if you remember that patients affected with anhidrotic ectodermal dysplasia exhibit mutation either in here or in here, we demonstrated that the patients that exhibit mutation in here lose affinity for both linear chain and lysine 63 chains, while patients carrying mutation in the zinc finger only lose the affinity for lysine 63 polyubiquitin chains. This is basically how we think this complex do form. So this in green is a linear diubiquitin which contacts the NOA domain at 2 sites, in white and in red, while lysine 63 chain, indicated in brown, are able to contact the NOA domain only at 1 site, but are also able to contact the C-terminal zinc finger, therefore increasing the affinity. What is purely hypothetical in this cartoon is first the linker domain, the structure of which is unknown, and it's probably unstructured at least until it meets polyubiquitin, and the number of ubiquitin moieties between the NOA and the zinc finger. I've drawn 4 of them, but they could be any number between 2 or 150 or whatever you can imagine so far. We are trying to get the crystal structure of the entire complex, but so far we have no information about that. So why did I tell you all this? Because, again, about at the same time we were interested in understanding what the role of linear and lysine 63 chains on NF-kappaB activity. Jean-Laurent identified 3 patients which exhibit a new type of syndrome associating in a rather unusual manner immunodeficiency, autoinflammation, with something even more strange, which is called amylopectinosis. And these 3 patients had a mutation in one of the subunits of the LUBAC complex, one of the regulatory subunits. So this basically a summary of characterization of the patients. Pyogenic bacterial infection, autoinflammation, this is one of the patients, which normally doesn't very often go together with immunodeficiency. And what is amylopectinosis? Basically, it's the accumulation of abnormally branched glycogen into muscle cells, including the heart, giving rise to myopathy and cardiomyopathy. And to the best of my knowledge, I don't think this has ever been associated with immunodeficiency so far. So this genetic analysis of the patients, 2 of them inherited a deletion from one parent, a stop codon for the other parent. The 3rd patient inherited a frameshift mutation at the beginning of the coding region that gives rise to premature stop codon. And Western analysis demonstrated that indeed those 3 patients lack the Hoyle subunit. But what's more interesting is that they also have a reduced amount of the 2 other components of the complex, HOIP and sharpin, which means that Hoyle is probably required for the stabilization of the entire complex. So, as I mentioned, the Lübeck complex is probably involved into recruitment to the receptor of the signalosome. So what we did was to test for the recruitment of the NEMO to both TNF and IL-1 receptor. So this is a crude lysate demonstrating the appearance of ubiquitinated NEMO after 5 and 15 minutes of TNF. But the interesting data is where you immunoprecipitate the entire complex from the TNF receptor. What you observe is a very strong enrichment in ubiquitinated NEMO, but possibly the most interesting information is that there's basically no difference between the patient and the control cells, contradicting most of what has been published before, saying that linear ubiquitin is critical for TNF signaling. So of course the question we ask is, does this correspond to a real signaling defect, or it's just some type of biochemical artifact? Well, before doing this, we tested IL-1, and where the IL-1 situation was very different, in the sense that there were basically no recruitment of NEMO to the IL-1 receptor in the patient cells, and no recruitment of the Lübeck complex, or at least what's left of the Lübeck complex. So in order to confirm that it indeed corresponds or not to a difference in TNF and IL-1 signaling, Jean-Laurent's collaborator, Virginia Pasquale at the Baylor and Damien Chosabel in Seattle, ran a transcriptome analysis of fibroblasts derived from Hoyle-deficient patients after TNF and IL-1 treatment, and compared to MyD88 and NEMO-deficient cells, and of course to control cells. And actually the result confirmed what we observed in the recruitment experiment, which is basically the 2 patients showed almost no, or at least minimal interference with the normal profile exhibited by the control cells in response to TNF, very different from a NEMO-deficient cell, and of course similar to MyD88 patient, which have no reason to have an interference with the TNF pathway. And on the other hand, the IL-1 situation was very different, in the sense that the 2 patients had a very strong defect in the IL-1 response, 3 controls, 2 patients. It looks like a bit like the NEMO-deficient or MyD88 deficient cell, except for a subset of gene essentially encoding chemokines around here. So for fibroblasts, the conclusion is that oil-deficient fibroblasts have an almost normal TNF response and a very strongly reduced IL-1 response. But the surprise came when we studied the blood cells. PBMC here, I don't show you all the data because actually we don't have all the data yet. But I just want to show you the IL-1 response, which is, I think, striking. The IL-1 response of Hoyle-deficient PBMC was actually the opposite of what you could expect from what is found in the fibroblast. In fibroblast, it's almost completely down, while in PBMC, that's the opposite. You get a hyper response to IL-1 of a large number of transcripts. And in particular, those subset of genes on the bottom parts of the heat map are stimulated 5 to 10 times more than the control cells by IL-1. And this gene include a number of chemokines, cytokines, plus a number of genes which are more or less connected with the inflammatory response, including COX-2. So that's where we are at the moment. So this is a bit perplexing. And of course, we wanted to know which cells within the PBMC were responsible for the hyperresponse. And so far, the only cells that we've been able to test are EBV transformed B cells that show that in response to IL-1 on CD40, the NF-kappaB response is completely down, which means that other cells within PBMC are probably responsible for that hyperresponse. NK, T cells, monocytes. We have to determine which cell or cell combination is responsible for this response. So that raises, of course, a very large number of questions. I've listed some of them here, but I guess you can add many others at the bottom of the list. Why is the TNF phenotype different from IL-1? Why do fibroblasts behave differently from PBMC? What is the actual role of flubac in the NF-kappaB pathway? Because it's been postulated that it only plays a role in the NF-kappaB pathway, but it's by no way been absolutely confirmed. And last question, among others, does Hoyle have a function outside of the Lubach complex? We know that sharpin has a function outside of the Lubach complex, so Hoyle may very well have a function outside of the complex that would explain amylopectinosis. So I'll stop here. And the last slide show you Jean-Laurent and Capucine Picard in the Paris group, plus postdocs at the Rockefeller, and Emmanuel Laplantine in my lab who done all the biochemical experiments. Thank you for your attention.